So in this video, I'd like to talk about DNA replication, the process by which DNA is copied. So as we noted, in our simplest possible organism, each cell needs a set of instructions. So that means in order for this single cell to duplicate to make two cells, that we have to copy all our sets of instructions so that when we divide, that we have two cells, each of which carries a full set of instructions. That is, DNA must be copied in order for cells to reproduce. So if we look at our double-stranded DNA, you may have already noticed that actually we only need one strand to figure out what's going on. So if we suddenly lost one strand of our DNA, we could reconstruct it because we know from our rules that there's a G, so it must be a C, there's a T, so it must be an A, and then we know this one's T, this one's G, this one's C, and so forth. And it turns out to be the case that DNA replication takes advantage of this fact. So the way that DNA is replicated is that the two strands are separated, and then each strand has its partner created nucleotide by nucleotide by adding on the nucleotides that we know must go with their partners. So we just use our rules to reconstruct the two strands from each single strand. So if we think about that, that would just look like this. We start with our double strands, and then we separate them. And then each one goes and has its extra strand added. Great. So we're done, right? End of video. Well, it turns out it's a little bit more complicated. So DNA molecules are huge. So in humans, an average DNA molecule is a chromosome, which means it has 10, or it doesn't. So each chromosome has on average about 100 million nucleotides. So that means if we were to pull this apart, we'd have single-stranded DNA, which is pretty unstable and reactive, and we'd have a huge strand of single-stranded DNA, or two whole strands. So having the whole thing single-stranded at once, this would be dangerous. This would be reactive, it would be unstable. The other thing is DNA, as we know, is a helix, and so to separate the whole strand, actually you'd have to pull it apart, you'd have to twirl both ends of the strand a huge amount. So it turns out that it's done in pieces, and so let's look at that. So instead of separating the whole thing, what we're going to do instead is just separate a little bit of it, and then we're going to replicate a little bit of it, and then we're going to move along in one direction, and add a little bit of it, move along and add a little bit, move along and add a little bit. So that means we do it piece by piece. And we're going to call this this moving part, this fork, the replication fork. So, great, we're done, right? Well, actually, no, it turns out there's one other complication, and actually you already know enough to figure out that complication. So here I've just blown up our little replication bubble a tiny bit. So let's work this out. So let's say that the bottom strand is going 5' to 3', 5' to 3', 5' to 3' for each nucleotide. It's going 5 to 3 to the left. That would mean that the top strand must be going in the opposite direction. And so now we can figure out our two new strands. Well, they're paired with strands in the opposite direction. So this one must be going to the right, this one must be going to the left. Okay, so now let's think about this. So, okay, we open up, we start our replication, and we replicate little bits. And then we move the replication fork to the left. Well, now on the top, we can add each nucleotide, each one adding to the 3' end of the strand. But that means that if we look at the bottom, that's the wrong end, right? That's the 5' end, as I've drawn it. And so that means we can't add nucleotides, because as we learned, nucleotide strands only grow on their 3' end. Okay, so then what that means is that actually the way it works is like this. Okay, so we open up a little bit of bubble, and then we synthesize both of the DNAs, and then we extend the bubble to the left. And now what we're going to do, so the top strand is going to add nucleotides to the to the strand that the same strand that we were creating before. But on the bottom, we're going to need to actually start a new strand on the left and keep going until it joins the strand to its right. And in fact, the top one's going to be a little bit faster, so it's going to get going before the bottom one does. So we're going to call the top one the leading strand because it goes first, and the bottom one the lagging strand because it lags a little bit. And so that means if we were to look at a region of it, the top strand is going to be this one single strand. I've colored it in one color. Whereas on the bottom, actually the bottom is going to be synthesized in these little chunks. Okay, right, we got it? Great. Actually, no. Probably by now you know there's probably one more thing. So DNA polymerase, the machine that is actually doing this, that is creating these new DNA strands, it can extend a strand, but it actually cannot start a strand. So that means if we are here and we need to start a nucleotide there, that we can't, sorry, we do the top, but on the bottom, we can't start this with DNA polymerase, because DNA polymerase cannot place the first base. DNA polymerase cannot place the first base. Okay, so there you go. You got that memorized. The great rhymes. But an RNA polymerase, which is called the primase, can place the first base. I mean, it can place an RNA base. So it's a little bit weird, but let's have a look at what this looks like. So that means that the first nucleotide is going to be added by this RNA primase. And so I've added this little O here to indicate that this nucleotide is an RNA nucleotide. Because remember, RNA has one extra OH group relative to DNA. So what happens then is that the primase adds the first RNA base and then another RNA base. Okay, so now we have bases placed there, and as we said, DNA can extend a strand. So it turns out that DNA polymerase can start with this little RNA primer, and then can add nucleotides one at a time. And so actually, if we think about it, that means that this blue thing, that actually it must also have started with in this case, two RNA nucleotides. And so then what that means is that when this orange strand is extended, eventually it hits this RNA strand. But we don't want RNA in our double-stranded DNA helix. And so what happens is that these nucleotides get removed, and then this gets extended, and then these two, though I haven't drawn it here, these two strands, so they're still separate strands, right? They need to be attached. And so they're ligated by an enzyme called DNA ligase. So what that means is that if we're looking at this bottom strand made up of different independently synthesized strands, that every place that we've got, like here, the end of this orange used to be the RNA beginning of the blue, and the end of this purple used to be the RNA beginning of the orange, and so forth. Okay, so here I've drawn something out, and so a main thing we want to be able to do here is to sort of identify how all this works by being able to label the different parts. So we want to be able to label the replication fork, the Okazaki fragments, leading lagging strands, RNA primers, where the RNA primer used to be, and the origin of replication. So it's a nice, this is a good thing to practice on. And so here's the kind of answer we're looking for. The lagging strand is this whole thing. The leading strand is this whole thing. The replication fork is the bit on the left that's moving. The origin of replication is where this all kicked off. We've got our ends of the fragments where the RNA primer used to be, our Okazaki fragments, and then our little bits of RNA. Okay, so one more thing. So here's some terminology, which we just went through, but here it is all written down. And so now we just have just one more thing to point out here, is that, sorry, the yo here was that I was supposed to add a little video for you, but in fact I'm going to do it some other place. But you can just imagine that I'm just saying yo. So another thing is to point out that the bits, these little bits of DNA, are not wicked short, like 9 nucleotides as I've drawn them. There's a lot of variation between species, but in general, RNA primers, the little RNAs, are between 8 and maybe 100 nucleotides, and the full synthesized bits, that is the Okazaki fragments, are between 100 to 2,000 nucleotides. You don't have to memorize that, but just to give you a sense, it's not just 2 RNA nucleotides and then 6 DNA nucleotides or whatever. They're longer than that. That was just for purposes of presentation. So the last thing that I want to add for this video is that the ends of chromosomes are a problem. So let's think about this. So if this is our double-stranded DNA, then the end of it, the end of this chromosome would look like this. So if we think about this, so if we have our replication fork, then for one strand that's fine because we can just draw it from left to right and it'll go all the way to the end. But what about this? This strand is a problem, right? Because we need to start here, but we need to start with an RNA, and then the only way the RNA is replaced is if we come in from the left with some DNA to replace it. But we can't do that because there's nothing over here to complement. There's no place over here to start. So here's what I'd like you to know about this. We call these ends of the chromosomes telomeres. And then I'd just like you to know that the solution is a trick. And this trick involves using this weird enzyme called reverse transcriptase, which is able to start a DNA strand, unlike DNA polymerase. And the other thing is that the other part of the trick is that telomeres have this sequence that is highly repetitive. So it'll be the same short sequence again and again and again and again. And what this means, if we have hundreds of copies of this short sequence, is that even if we make some mistakes, even if things aren't quite perfect here, if replication is a little sloppy with different numbers of repeats in different cells, it's probably going to be okay. In addition, it's part of this reverse transcriptase trick that I've decided we won't talk about here. But if you'd like to look it up more, I'll put a video on YouTube. Or I'll put a video on we'll put a link to a YouTube video on iLearn. Okay, so here are our main things that we covered in DNA replication. We've got the origin of replication and then the moving replication fork. We've got leading and lagging strands, RNA primers, Okazaki fragments, and DNA ligase that create the lagging strand, and then telomeres, the ends of chromosomes.